Hello and welcome. Today's topic is solute transport across membrane. I'm your host, Dr. Deepak Verma. So, what is the membrane transport? As we know that every living cell must acquire from its surrounding the raw material for biosynthesis and for energy production and must release into its environment the byproducts of the metabolism. So, So it is important to in and out of the material from the membrane. And we begin with this section by describing the permeability properties of protein-free synthetic lipid bilayer. Then we introduce some of the terms used by describe the various forms of membrane transport and some strategies for characterizing the protein and process involved. So why this transport is necessary? So here I am showing you a lipid bilayer which is devoid of the protein. Here lipid molecules can easily go dissolve in this lipid bilayer and cross this very easily, lipids and fatty acids. But for sugars, amino acids, water molecules cannot go inside the cell, and cell need these things for the metabolism. And also this waste product such as ammonia and salt and various other products, byproducts, they have to be removed from the cell, but they cannot cross this barrier, this phospholipid barrier. So that's why the membranes, this protein, proteins uh, become proteins are necessary to for this permeable. That's why this plasma membrane is known as the semi-permeable membrane. So all the cellular membranes, both plasma membrane and ordinary membranes, consist of a bilayer of a phospholipid in which other lipids and specific type of proteins are embedded. And it is this combination of lipid and protein that gives cellular membranes their distinctive permeability qualities. If cellular membranes were pure phospholipid bilayer, as I showed you earlier, they would be excellent chemical barriers, impermeable to virtue of all ions, amino acids, sugars, and other water-soluble molecules. In fact, only a few gases and small uncharged water-soluble molecules can readily diffuse across the pure phospholipid bilayer. But cellular membranes must serve not only as a barrier but also as conduits, selectively transporting molecules and ions from one side of the membrane to the other. So energy-rich glucose, for example, must be imported into a cell and waste must be the shipped out. In this figure, I am giving a virtual idea of how diffuse the protein-free lipid bilayer down in its concentration gradient. The rate at which it does so, however, varies enormously depending partially on the size of the molecule, but mostly on the its relatively soluble in oil. In general, the smaller of the molecule and the more soluble in its oil, the more it rapidly it will diffuse across the lipid bilayer. Small non-polar molecules such as O2, CO2 readily dissolve in the lipid bilayer and therefore diffuse rapidly across them. Small unchanged polar molecules such as water or urea also diffuse across a bilayer mm, will much more slowly. By contrast, lipid bilayer are highly impermeable to charged molecules such as ions, no matter how small it is. The charge and the high degree of hydration of such molecules prevents them from entering the hydrocarbon phase of the bilayer. Thus, synthetic bilayers are almost 10 to the power 9 times more permeable to water than to even such small ions as sodium ion and potassium is normal. Now we know that proteins are important for transportation. So now we move to the our section to the protein. So there are three main class of membrane proteins: transport molecules and ion across the cellular membrane. As I seen you earlier, very few molecules and no ions can cross a pure phospholipid bilayer at a pre-fill rate by simple diffusion. Thus, transport of many molecules into and out of the cell require the assistance of specialized membrane protein. Even in the case of molecules with relatively large partition coefficient such as urea, fatty acids, and certain gases such as CO2 and ammonia, transport is frequently accelerated by the specific protein because simple diffusion does not occur rapidly enough to meet the cellular need. All membrane proteins are transmembrane proteins containing multiple membrane spanning segments that are generally alpha helix. By forming a protein line pathway across the membrane, transport proteins are thought to allow the hydrophilic substance to move through the membrane without coming into the contact with its hydrophobic interior. Here we introduce three main types of membrane transport protein covered. So now we discuss about this picture. So the first is the channels. Channels permitted movement of specific ions or water down their electrochemical gradient, and while the transporter, which fall into the three groups: facilitated movement of the specific small molecules or ions, uniporter transport a single type of molecule down its concentration gradient, and co-transporter and symporters and antiporters. So they catalyze the movement of one molecule against to its concentration gradient. They are shown in the black circle. Drive by movement of one or more ions down in electrochemical gradients, which is shown as here with red circles. And the last is the pumps, used for the energy released by ATP hydrolysis to power movement of specific ions or small molecules in red circles here against their electrochemical gradients. Difference in the mechanism of transport by three main major class of protein account for their varying rate to the solute movement. So now we come one by one that channels transport mainly the water, specific ions, or hydrophilic small molecules across membrane down their concentration electrical potential gradient. Because of this process required transport proteins but not energy, it is sometimes referred as a passive transporter or facilitated diffusion, but it is more properly called facilitated transport. Now we move to the transporters, also called the carriers. They move a wide variety of ions and molecules across the cellular membrane, but at a much slower rate than channels. Three types of transporter as earlier: uniporter, again transporter, unitransporters transport a single type of molecule down its concentration gradient. For example, glucose and amino acid crosses the plasma membrane into the most mammalian cells with the aid of uniports. Collectively, channels and uniporters are sometimes called facilitated transporters, indicating movement down a concentration of electrochemical gradient. In contrast, the antiporters and symporters, coupled with the movement of one type of ion or molecules against its concentration gradient with the movement of one or more different ions down its concentration gradient in the same porter or different antiporter direction. These proteins are often called co-transporters because their ability to transport two or more different solutes simultaneously. Now we move to the this pumps, ATP powered pumps, simply we know called as pumps, are ATPs that are use energy of ATP hydrolysis to move ions or small molecules across a membrane against a chemical concentration gradient and electric potential or both. This process referred as the active transporter as an example of coupled chemical reactions. In this case, transport of ions or small molecules uphill against the electrochemical gradient which requires energy is coupled to the hydrolysis of ATP which releases energy. Here, always remember one thing, that it's against the concentration or the potential. So now we move to the next portion. There are two main classes of membrane transport proteins. One are transporter and channel. Like synthetic lipid bilayer cell membranes allow small nonpolar molecules to permeate by diffusion. Cell membranes, however, also have an to allow the passage of various polar molecules such as iron, sugars, and amino acid, nucleotide, water, and many cell metabolites. They cause synthetic lipid bilayer only very slowly. Special membrane transport protein transfer such solute across transmembrane. These proteins occurs in main form in all type of biological membranes. Each proteins often transport only a specific molecule species or sometimes a class of the molecules such as ions, sugars, or amino acids. Now we know that humans and similar mutations suffers from various inherited diseases that hinder the transport of the specific solute or solute class in the kidney, intestine, or other cell types. Individuals with inherited disease such as cystic CFTR, they cannot transport certain amino acids such as cysteine, and this is other thing from either to the urine from the intestine into the blood. The resulting accumulation of cysteine in the urine leads the formation of the cysteine stones in the kidney. All membrane. Now we come to the actually we let anyway come to the topic. All membrane transport proteins that have been studied in details multi pass transmembrane proteins, that is, their polypeptide chains transverse the lipid bilayer multiple times. By forming a protein-line pathway across the membrane, these proteins enable specifically hydrophilic solute to cross the membrane without coming into direct contact with the hydrophobic interior of the lipid bilayer. Transporter and channels are the two major class of the membrane transport proteins. Transporters, also called carriers or permeases, bind to the specific solute to be transported and undergo a series of conformational changes that alternatively expose solute binding site on the one side of the membrane and then on the other transfer of the solute across it. While the channels, by contrast, interact with solute to be transported much more quickly, they form continuous pores that extend across the lipid bilayer. When open, these pores allow specific solute to pass through them and thereby cross the membrane. For example, such inorganic ion appropriate size on sometimes is glycerol, ammonia, also and also sometimes water also. Not surprisingly, transport through channels occurs a much faster rate than the transport mediated by transporters. Although water can slowly diffuse across synthetic lipid bilayer, cells are dedicated channels proteins, water channels, aquaporins that greatly increase the permeability of their membranes to water. Later, sometimes we will discuss sometimes. So here we will show you transporter and channels difference between. And I thought that clearly. Now How we form the transport membrane transport. So all channels and many transporter allow solute to cross the membrane only passively. Passively means the downhill, means the concentration according to the concentration gradient. The process and this process is called the passive transport. In this case, the transport of a single uncharged molecule, the difference in the concentration on the two sides of the membrane, concentration gradient, drives passive transport and determine its direction. If the solute carries a net charge, however, both its concentration gradient and the electrical potential difference across the membrane potential influence its transport. The concentration gradient and the electrical gradient combine to form a net driving force, the electrochemical gradient. For each charged solute, we discuss some anytime, any other time. In fact, almost all the plasma membranes have some property to cross this. Which way I'm going to the electrical one, not I have to come back. So as shown in here, cell needs to be able to actively pump certain solute across the membrane uphill against their electrochemical gradient, such as active transport is mediated by transporter whose pumping activity is directional because of its tightly coupled to a source of metabolic energy. And as we know that ATP is the unit of energy in cell. So transmembrane movement of a small molecule mediated by transport can be either active or passive, whereas mediated by channels is always passive. Now we move to the transporters and active membrane transport. So first we will discuss about passive, then active. The process by which transports surfaces a solute molecule across the lipid bilayer resembles an enzyme substrate reaction. And in many ways, transporters behave like enzymes by contrast to ordinary enzyme substrate reaction. However, the transporter does not modify the transported solute, but instead delivers it unchanged to the other side of the membrane. Each type of transporter has one or more specific binding sites for its solute, means here the substrate. The transfer the solute across the lipid bilayer by undergoing reversible conformational change that alternatively expose the solute binding site first on one side of the membrane and then onto the other, but never on both sides at the same time. The transition occurs through the intermediate state in which the solute is inaccessible or occluded from either side of the membrane, as shown in the figure, middle one. And when the transporter is saturated, means when all the solute binding sites are occupied, the rate of transport is maximum. This rate referred as Vmax and its characteristic of the specific carrier. Vmax measures the rate at which the carrier can flip between the conformational states. In addition, each transporter has a characteristic affinity to its solute reflected in the Km of the reaction, which is equal to the concentration of the solute when transporting rate of half is the maximum value. As with enzyme, the bindings of solute can be blocked by either competitive inhibitor or non-competitive inhibitor. As we discussed shortly, require only a relatively minor modification of the model showing here in order to pump a solute uphill against the electrochemical gradient. Cells carry out such active transport in three main ways: coupled transporter, ATP driven pumps, and light redox pumps. These are the three ones, and they actively. Now we comes to the coupled transporter. First, one by one. Coupled transporter harness the energy stored in concentration gradient to couple the uphill transport of one solute across the membrane to the downhill transporter of another, while ATP-driven pumps coupled uphill transporter to the hydrolysis of ATP. And the last is the light or redox-driven pumps, which are known in bacteria, archaea, mitochondria, and chloroplast, coupled uphill transporter to an input of energy from the light as a bacteriorhodopsin or form a redox reaction as a cytochrome C. And up to here we are discussed about the small molecules, and what about the large molecules, how they are transferred from exterior to interior or interior to exterior. So this is not from, actually this is not studied in transport system. These are actually the endocytosis. Endocytosis inside, to go inside the cell. Right? There are two types, two forms: phagocytosis and pinocytosis. Phagocytosis is the for cellular eating. Is we have used this term in various immunological, and pinocytosis is a cellular drinking. Is something evidence like in amoeba. Here I am showing you phagocytosis that fused with the lysosome for digestion and used for its metabolism. And this pinocytosis actually is non-specific process, and sometimes it's receptor mediated endocytosis. It's triggered by the molecular signal. And endocytosis, as we know that from releasing something from outside the cell. So thanks for listening. I hope you understand this all the things. Okay.